Look at me looking like Mr. Anderson. No, not really, but um this video I want you to go and watch 2 times. Once I want you to sit and watch it straight through. The other time I want you to watch and pause and take notes about the relationship between amino acids, how they make up polypeptides, and how polypeptide chains make up a protein and how that's related to ribosomes. Okay. So that's kind of your assignment. And you will need to be able to hear it. So that's an issue. Maybe it needs to be done at home. But you got to add on to that. So in talking about proteins and enzymes, we need you to know that proteins are made up of amino acids. They are a polymer of amino acids that are formed together, what's called a polypeptide chain. But that polypeptide chain, it can be like you see this primary structure that's listed here on the screen. It tells which amino acids come in what order. But then it begins to fold up. It creates beta sheets, an alpha helix, and then there's tertiary structure and quaternary structure as more than one complex may join together to create enzymes that have specific molecular jobs in the body or structural proteins. Enzymes are proteins. Not all proteins are enzymes, but all enzymes are proteins. And enzymes are really like the chemical reaction vehicle for many of the things that take place in your body. Proteins are made up of amino acids. This is a amino acid structure. It's the building block of proteins. So you can see there's some different components. You see hydrogen. I know you see oxygen and you see carbon. Voila, that is very familiar. Then beyond that, there's nitrogen involved. And there's a functional group here, this R group. And the R group has a lot of different features. And that, I think that's what Mr. Anderson talks about in the Bozeman video, is kind of like the chemical or the charge features that are different in that functional group, that R group, and it can really like change the way that particular amino acid functions in the body. Proteins have a lot of different functions, but one of the main one is structure and support. So an example of that would be keratin. Keratin is a protein that you're probably pretty familiar with. It's found in hair and hooves, feathers, claws, skin, and it is an extremely strong protective molecule that like if I knock my hand against something hard, it doesn't just like blow a hole in my epidermis. That the proteins are able to like withstand a lot of forces pulling on them. So there's a lot of tensile strength. And as those cells become keratinized and full of keratin protein, they're bonded strongly together. They are resistant, not waterproof, but they're resistant to water and they're resistant to abrasion. They also are stretchy. And so that can help to kind of keep everything where it needs to be. You have this really incredible chemical capacity within your body. Like structurally, your body is just a bag of chemicals that has an amazing shape and stays in that particular shape. Collagen is another biological protein. It is the most abundant protein in the body and can be found in bones, muscles, tendons, anywhere where there's some kind of scaffold for support or cells are built around that. Elastin is another example of a protein and this allows the protein tissue to keep its shape so it can be stretched and it will go back. Myosin and actin are other essential proteins that are the functional capacity for muscles. So you can see this myosin filament and the actin filament. These actually kind of like ratchet along each other, and that's what makes muscles shorten, so that you can see in the top section you have what's called a sarcomere, which is a section of muscle, and those filaments are apart. That muscle is relaxed, and then when those proteins interact with one another and each protein moves in tight on each other, that muscle contracts. So the building blocks of proteins are called amino acids, and you can see that you've got this chain, this long chain of polypeptides and or of amino acids. This is the long chain is what we call the polypeptide. And each amino acid has a unique chemical structure. It also has unique physical properties and chemical properties in therein, and they're named. Each one is named differently. So structure, so kind of like you get like different chunks that come in there together. That's just showing how complicated proteins are. So it's not just the strand of amino acids, but it's also the way they interact with each other. And so there's a certain like charge on one, or like some are attracted to each other, some will repel each other, and that gives the capacity for that polypeptide chain to fold in multiple ways. And that, I think, is about where, oh, no. Okay, we're going to talk about ribosomes. Ribosomes are where polypeptides are built. So a ribosome is ironically a protein itself, but it also helps in the process. It reads the messenger RNA, and then as it reads messenger RNA, there's like the RNA comes in kind of a set of three, and there's these, not kind of, it comes in a set of three, and it reads three of those coded what's the word? Hang on, deoxyribonucleic acid, nucleic acids. So the nucleotides there, 1, 2, and 3. So either, adenine, adenine, uracil, guanine, or cytosine, which is part of RNA. It'll read those and does the complementary match with the tRNA, the transfer RNA, and the ribosome will actually like string together amino acids that are coming in on a transfer RNA. It's a little machine, like it's a little factory. And the factory parts are the transfer RNA brings the amino acids, they're all separate, and then strings the amino acids together. So the ribosome is an organelle that is specifically involved in building a string of amino acids into a polypeptide, but then the polypeptide is going to fold in on itself just because of the structure that it has in terms of like what order those amino acids are put in together. So the big question at the end of this is, how are amino acids, polypeptides, proteins, how are those all related? And how are they related to ribosomes? So you can see the answer here. Amino acids are strung together to build polypeptide chains. These chains eventually fold to become a functional protein. And ribosomes are where this actually takes place in the cell. Ribosomes are the site for protein synthesis. All right. And now on to a little bit about enzymes. Like I said before, all enzymes are proteins. Not all proteins are enzymes. But enzymes are the workers, basically. This is what does the work in your cell, and it has to do with interaction and this of a substrate with the enzyme with a particular substrate, and it helps to catalyze a reaction, basically. Enzymes have active sites that will bond with certain molecules according to the shape of the molecule. So we talked a little bit about like lactose. Lactose is a sugar that requires an enzyme in our body, lactase, to break it down. If you don't make lactase, you're not going to be able to break down lactose. You can take lactade, which is lactase enzyme that mixes in your digestive system and breaks down lactose for you. That's an easy fix, well, to some degree. Enzymes are catalysts. They help a reaction happen easier. Basically, so like in this example, this is an enzyme for called sucrase that's going to break apart sucrose. So the substrate, which is the material that's going to be acted on by an enzyme, the substrate is called sucrose. That's a sugar, right? Table sugar. We talked about that. And your body has an enzyme called sucrase. Like I'm pretty sure it's even in your spit, which I might be wrong, but I'm guessing. If not, it's in your digestive juices. It binds with the substrate with sucrose. That activates the enzyme, and then we add some water into that. The substrate is then converted into glucose and fructose as it's broken down. It needs water to go into it because in order to get that bond to break apart, there has to be a hydrogen and a hydroxyl group available to satisfy, because remember, dehydration synthesis gave off a water. So in order to break apart a molecule, you need water to go in to make those new molecules stable. Otherwise, they would, be without a bond on one side or the other. This enzyme can then be used again for the substrate. So you can add more substrate. The substrate will react with the enzyme. As long as there's water available, it will continue to break down sucrose. Enzymes work by decreasing the amount of energy that a chemical reaction actually needs to occur. So if a chemical reaction takes a high amount of free energy, this is why cooking substrates often will help break them down. You can cook sucrose, or you can use an enzyme. Or you can use an enzyme in your digestive tract, and that would require a little bit less free energy in order to get the reaction to take place. Enzymes have a few properties that you need to know about. They are specific to one substrate only. It is like what we refer to as the lock and key analogy, that a substrate has a particular shape and that the active site of the enzyme fits that shape. So they're very, very closely related. The name of an enzyme typically ends in ase. Like I said, lactose is the sugar, lactase is the enzyme. Sucrose is the sugar, sucrase is the enzyme. Lipase, catalase, lactase, like I said. And got one more video for you on enzymes. So you go ahead and click off of this, find that video, add to your notes, and that gets us through proteins. And maybe that gets us through the end of the presentation. I think so. Bye, guys.